Hi everyone, welcome back. The learning goal for this video is for you to be able to describe the attractive forces between ions, polar covalent molecules, and non-polar covalent molecules. And this knowledge you'll gain from watching this video and practicing problems will help you understand better the attractive forces that play a role in the body. We'll discuss this at the end of this video, but for example, the protein shape, the three-dimensional structure of this protein here is stabilized by attractive forces such as hydrogen bonding between functional groups of side chains, which we refer to as R groups on the amino acids, causing it to twist and bend into a specific three-dimensional shape. And then that shape determines the function of that protein in our body. So in the previous video, we discussed how to determine if a molecule is polar or nonpolar overall. So you'll use that knowledge from a previous video. If you have a polar molecule, they will exert attractive forces called dipole-dipole attractions. They possibly will form strong dipole attractions called hydrogen bonds, but what's needed for hydrogen bonding to occur is to have hydrogen atoms already attached to fluorine, oxygen, or nitrogen, which are the three most electronegative elements on the periodic table. So we see here in this case, this hydrogen is bonded to nitrogen, but this is not the hydrogen bond. It's the interaction between this hydrogen and the lone pair of a nitrogen in a neighboring molecule. So the hydrogen bonding is this ... here. And although bond is a misnomer, it's not an actual bond, it's an attraction. So they're not necessarily glued together, not as strong as these covalent bonds, but once again, it's just an attraction. Here we see hydrogen chloride. This doesn't have nitrogen, oxygen, or fluorine, so this would not participate in hydrogen bonding, but rather this would just be classified as a dipole-dipole attraction. And hydrogen bonds are the strongest force between molecules and play a major role in the shape of DNA and also of proteins. Now, dispersion forces are weak attractions between nonpolar molecules. So for example, if you have a nonpolar molecule and I ask you what's the dominant attractive force here, you would say dispersion. These are temporary dipoles that are developed when molecules bump into each other. So I like to think of this if you've ever done bumper cars. So, you're driving a car and then someone comes and bumps up against you, and that interaction causes like this temporary like disruption or, this temporary dipole in this case here. And this is a dispersion force. That's weak, but it does make it possible for nonpolar molecules to form liquids and solids rather than just being gases at room temperature. Now, the reason why we're talking about these attractive forces is that it really impacts not only the structure and function of proteins and DNA, for example, but it impacts the properties of different compounds, such as melting point. So melting points of compounds are related to the strength of the attractive forces between molecules or compounds. The melting point is lower due to weak forces, such as dispersion forces. So molecules that only have dispersion forces will have low melting points. And molecules that have stronger attractive forces, such as hydrogen bonding, will have higher melting points. And then if we're talking about ionic compounds, which is even stronger than hydrogen bonding, those are very strong attractive forces between ions. Those compounds have the highest melting point. So the trend we see here, and by the way, attractive forces I will refer to more so as intermolecular forces. Intermolecular forces, I like to abbreviate that as IMF. Your textbook is referring it more to attractive forces. They're synonymous with each other. So as the intermolecular force or attractive force increases, the melting point also increases. It's just harder to pull those compounds apart if they're really attracted to each other. So here are some examples of melting points of different substances. So for example, ionic compounds have a very high melting point compared to, for example, the non-polar molecules, which only have dispersion forces, have very low melting points. And you can see hydrogen bonding intermolecular forces or attractive forces are stronger than the dipole-dipole attractive forces alone. Here's showing you a comparison of like bonding and attractive forces. So when we're talking about intermolecular forces or attractive forces, we're primarily going to look at hydrogen bonding, dipole-dipole, and dispersion. Remember, hydrogen bonding and dipole-dipole is for polar molecules overall, and then dispersion nonpolar. These are the attractive forces. These are the different types of bonding. So covalent bonding. So notice how these are just ... That's the attractive force, but a covalent bond is a sharing of electrons. That's stronger than an intermolecular force or attractive force here. And then the strongest bonding is ionic, because it's such a strong electrostatic attraction between positive and negative ions here. So go ahead, pause the video, try the study check on your own, and then check your work with me when you're done. All right, we are going to identify the main type of attractive forces: ionic bonds, dipole-dipole, hydrogen bonds, and dispersion forces. So the first thing I like to do, if I have a list like this, it's easy to determine the ionic compounds. So I go through and just see, like, are you ionic or covalent? I'm just walking through. And so if I look through these here, the only ionic compound I see is potassium chloride. That's the metal and non-metal. So immediately I would just say this is ionic. The rest are covalent. So now we've narrowed it down. Then I like to go through and see, like, are there any obvious non-polar compounds, covalent compounds? And if I walk through, bromine is non-polar because the electronegativity difference would be 0. So because it's non-polar molecule, then its attractive force would be dispersion. And that's the main type of attractive force it would experience. All right, nitrogen trichloride here. If we draw the Lewis structure, sometimes you have to go through and do the Lewis structures and determine the polarity. And the nitrogen chlorine bond is indeed a polar covalent. This is a trigonal pyramidal geometry. So the dipoles will not cancel out, and overall this molecule will be polar. Once again, we did that determining if a molecule is polar or non-polar based on Lewis structures and molecular geometry in a previous video. So if you're struggling with this study check here, then definitely email me, schedule an appointment with me, and we can work on this together one-on-one. Since this is polar, there's no hydrogens, then the only attractive force it can be is dipole-dipole. Water, we know, is a polar molecule. We worked on that in a previous video. We've seen that it has bent geometry. And it has hydrogen attached to oxygen. So we know that it can participate in hydrogen bonding with other water molecules. And so its main attractive force is hydrogen bonding. Because hydrogen bonding, remember, is when a hydrogen is attracted to a nitrogen, oxygen, or fluorine in another compound. But that hydrogen has to be attached to one of those electronegative atoms for that to work. So once again, if we were to draw another water molecule, the hydrogen bonding is not the OH, but rather the hydrogen bonding is interaction between this hydrogen in one molecule and the lone pair of the oxygen in the other. That's the hydrogen bonding attractive force. We've worked with ammonia before. It has trigonal pyramidal geometry, definitely polar covalent bonds. Those dipoles don't cancel out. And so therefore, overall, this is polar. And then we check, yes, there's hydrogens. Yes, the hydrogen's attached to one of these elements, the nitrogen in this case. So the dominant main attractive force here is hydrogen bonding. All right, so coming back to the example we saw on the first slide. So proteins are biological molecules with many different functions. They are needed for structural components such as cartilage, muscles, and hair, the formation of enzymes that regulate biological reactions. Myoglobin and hemoglobin are proteins which transport oxygen in blood and muscle. So proteins are composed of building blocks called amino acids, and every amino acid has a central carbon atom bonded to an ammonium group, which is from an amine, a carboxylate group, which comes from a carboxylic acid, hydrogen, and an R group. This is like the side chain, which is unique for each amino acid. So in this case, this is a methyl group here. So several amino acids have R groups or side chains that contain different functional groups, like an amide group, hydroxyl, carboxyl, which is ionized into a carboxylate in the body, an amine, which is ionized into ammonium into the body. And you can see here, this is what serine looks like, and this is its R group. This is what asparagine looks like, and this is its R group, and lysine and aspartate. So there's many different types of amino acids with different R groups. And the primary structure of a protein is the sequence of amino acids. It's the sequence of amino acids that helps us determine the protein's function. So amino acids in a protein are linked by a peptide bond. So this is an example of a peptide bond here. And that's when the carboxylate of one amino acid links up with the ammonium group of the next amino acid to form a peptide bond. Proteins then have a higher level of structure that's determined by the attractive forces that we just talked about between the amino acids. So when hydrogen bonds form between a hydrogen atom and a nitrogen, an NH group, and the oxygen of the C double bond O group, the protein forms an alpha helix. So we can see here hydrogen from an NH group, hydrogen bonding to an oxygen of a C double bond O group all throughout. And it's these interactions that cause the alpha helical structure of this protein here. Hydrogen bonds can also form between polar side chains of the amino acids and the polar water molecules in the external aqueous environment. So aqueous just means it's in water, and our bodies are mostly made of water. And so those proteins can also form hydrogen bonding with the aqueous environment. And so once again, these attractive forces you just learned about hold the protein in a specific shape. And we can see dispersion forces, ionic bonds between here and here, hydrogen bonding. So all of this is occurring to form the protein-specific shape so the protein can do its function in the body. All right, that wraps up this unit. So let's review over the concept map together. So we learned about ionic and molecular compounds. We spent a lot of time learning how to name them. We learned that ionic bonds are generally formed between metals and nonmetals, specifically between cations and anions that are positive and negatively charged respectively. And that we use charge balance because ionic compounds always need to be neutral to determine, the formula for the ionic compound, or if we need to use the charge balance to figure out the charge of a transition metal. And so that's all about ionic compounds there. Molecular compounds we learned contain covalent bonds, are between nonmetals. They can be classified as polar or nonpolar. We use Lewis structures to help us understand the structure of the molecule, but VSEPR theory determines the three-dimensional molecular shape. And with our knowledge of electronegativity and shape, we can determine the overall polarity of the molecule, which then will help us identify the dominant attractive forces or intermolecular forces for that molecule. And what we're looking at here is dipole-dipole, hydrogen bonding, and dispersion forces specifically. And that can impact not only our biological molecules such as DNA and protein, but also properties of those compounds such as melting point. All right, thank you for watching. And as always, email me with any questions.